Good day, everyone, and welcome to the session from species from specimen to sequence. This is an extremely important topic, maybe underappreciated until the advent of long read and long range technologies. So we're going to hear from a very bright selection of speakers about this topic. I'm here with Kirsten Ove, who is the lead of the production genomics for the Tree of Life at the Wellcome Trust Sanger Institute. Before we get started, just a couple of announcements. There are several posters associated with this session about specimen to sequence. So we encourage you to go and check them out on the portal. There are also some short videos associated with them. And please also, all the speakers and all the talks will be one after the other. So if you have a question, and we encourage you to ask questions, please make sure you put the name of the speaker in front of your question. So the first speaker is going to be Dr. Anthony Schmidt, is the Senior Vice President of Science at Arima Genomics. Is going to talk about chromosome spanning scaffolding, de novo assembly, and 3D genome analysis using Arima high coverage. Then we will have Jennifer Balacco, who is a lab operation lead at the Rockefeller University, talking about benchmarking ultra high molecular weight DNA preservation methods for long read and long range sequencing. Then we have Nancy Holroyd, who is a sample and partner relationship manager at the Sanger Tree of Life program. And her talk is going to be about supporting sample flow to feed growing Tree of Life. Then we have Dr. William Crowley from the Department of Zoology of the University of Oxford. His title is Sample to Sequence, Genome Sample Acquisition at Wyndham Woods. And finally, last but not least, Dr. Zemin Ning, also from the Sanger Institute, is going to talk about quality control of long-range NGS read from Hi-C, 10x, and haplotyping platforms. So thank you for being here with us today, and let's start with Dr. Andrew Schmidt.
Welcome, everybody. Thank you for attending the short oral presentation from Arima Genomics. My name is Anthony Schmidt, and I lead R&D at Arima. Today, I'll be briefly talking a little bit about the company and our technology platform at Arima, taking a deeper dive into sample preparation, touch a little bit on bioinformatics, and then wrap up with some concluding remarks. So to start, a little bit about the company. Arima is a biotech company based in San Diego, California, here in the US, aiming to accelerate the understanding of genome sequence and genome structure and the role of those two components in health, disease, and species evolution. And we facilitate access to this high quality sequence and structure information via kits and services. Here's just an image of our kit that we launched back in 2018, really initially designed to capture the three-dimensional structure of genomes at a genome-wide scale to enable genome assembly, as well as the analysis of genome structure and function. And since then, we've launched a couple different targeted versions of chromatin conformation assays that I won't have a chance to talk about much today. But more recently, last year, we launched high coverage Arima Hi-C, really with the genome assembly application in mind, which I will talk about more today. So a little bit about the core technology here. Here is just an overall workflow of the high coverage Arima Hi-C technology. I won't have time to go through this in detail, but one of the unique features about this is that we digest the genome in situ with a cocktail of 4 restriction enzymes. And ultimately what that enables is improved coverage uniformity of the genome, because the coverage uniformity is really dictated by the location of those cut sites. So for example, if you look at the coverage uniformity in humans and you analyze the coverage uniformity of the 4 enzyme chemistry shown here in purple is more uniform than our original sort of version 1 chemistry here in gray, which is also more uniform than some of the first generation Hi-C protocols from academic labs, which you can see is far less uniform. So that's one unique feature. This technology is utilized by the Darwin Tree of Life Project, the Vertebrate Genome Project, the Ag100 Pest, and many others. And really, I think the core benefits of this proven ARIMA workflow is really the reproducible and robust chemistry. I think that's one of the things that we're most proud of here, and how the technology just works in many different diverse tissue types from different sample sources. And by works, what I really mean is maximizing the capture of long-range intrachromosomal linkages that are effectively the scaffolding signal, while minimizing the interchromosomal linkages, which are effectively the scaffolding noise. And I should also point out that this technology is compatible to automation. We have some data internally on the Agilent Bravo. We know that some of the higher throughput users of this technology are also implementing automation. Now, taking a deeper dive into sample prep, this is just a general table of some of the validated sample sources like mouse and human that you can see over here on the left, and other vertebrates, as well as arthropods, parasites, nematodes, plants, fungi, algae, etc. Also many different sample types like whole blood, fresh or frozen animal tissue, or plant tissue. And then over here on the far right is just some of the general recommended sample amounts, such as 50 milligrams of frozen animal tissue, one insect, or even a partial insect, blood, and low input protocols are available for these different sample types. And additional evidence of this comes through publication that have used ARIMA technologies in humans, vertebrates, arthropods, nematodes, parasites, and plants. One of the things that we've been thoughtful about is really trying to establish a set of optimal sample collection and preservation methods that are amenable to Hi-C. So a couple years ago, we collaborated with Olivier Federigo and his team at Rockefeller to really achieve this goal of establishing optimal sample collection and preservation techniques. And just a little bit of data from this paper, which is now on bioRxiv, is we took zebrafinch blood, and it was collected and immediately flash frozen in 3 different sample preservation reagents, either no preservative at all, 95% ethanol, or DNA guard. It was held for 6 hours and then frozen in those same preservatives, or he was even kept for 1 week at room temperature and then frozen in those preservatives. And what does that data look like? Well, because this is done in zebrafinch and we can map the Hi-C data back to a chromosome scale reference, we can approximate the signal, here being the long-range intrachromosomal linkages shown here in dark purple. And we can also approximate the noise, which is the interchromosomal linkages shown here in light blue. And in the fresh frozen sample, as well as the 6 hour at 4C, you can see that no preservative and 95% ethanol works really well to capture the signal in the data and minimize the noise, whereas the DNA guard reagent immediately dilapidates the 3-dimensional architecture of the genome, and you get lots of this sort of noise signal in the data. I think what's most impressive here is that even out to a week, sitting in ethanol at room temperature, you still have a high capture of signal and a very limited amount of noise. We've also done this in tissues, and I would just divert you to the publication to see the tissue data as well, which actually turns out to be quite similar to the blood in terms of the optimal sample collection and preservation techniques. Lastly here, just a little bit to touch on bioinformatics. I wanted to start really with our philosophy here, which is that we support open source data analysis tools, and we have a very hands-on technical support team, which will help with things like pipeline installation, pipeline implementation for some of those widely used open source tools for genome scaffolding and phasing. We will share restriction cut site information or other information that's really necessary to use those tools. We'll help with data quality interpretation. We'll take a subsampled set of maybe the top 1,000,000 reads from your data set, run it through these pipelines ourselves, and share those QC analyses with you, and really try to take you through that whole process of getting the pipelines installed through analyzing and interpreting your data. The second thing that I wanted to mention is that we have a history of partnering to really help facilitate the development of new bioinformatics tools. So back in 2009, we had a wonderful partnership with Adam Philippi and Sergey Koren at the NIH to help develop SALSA2. A year later, from Romain Kojel's lab in France, the Instagrail scaffolding algorithm. And most recently, we've had a wonderful collaboration with Hong Li, Shilpa Garg, and George Church for the development of the DIPASM pipeline for chromosome scale haplotype resolved assembly of human genomes. So with that, I would just like to wrap up again by reiterating a few key points. The first point being ARIMA Hi-C technologies are established research tools with proven performance in terms of data quality and, importantly, the robustness of this chemistry across a range of sample types and quantities. ARIMA supports and helps develop open source bioinformatics tools for genome assembly. One thing I didn't get a chance to touch on very much is that ARIMA technologies also enable the study of genome structure and function following genome assembly. So once you have a genome assembly in place, to analyze the function of the genome and how the three-dimensional structure relates to the function. And our tech support team will help you really from sample prep through bioinformatics support. So I'd like to thank you all for your attention, and that's it.
Hi there, my name is Jennifer Blocco. I'm from the Vertebrate Genome Lab at the Rockefeller University. We're part of the Vertebrate Genomes Project, or VGP, and today I'll be presenting on some work done by the sample prep group of the VGP, looking into preservation methods for how to store a sample for ultra-high molecular weight DNA extraction for long-read sequencing. So as a little bit of background on the Vertebrate Genomes Project, we're an international collaboration of scientists with the ultimate goal of generating chromosome-level reference genomes, near error-free genome assemblies of all extant vertebrate species. To do this, we implement long-read data such as PacBio HiFi, Bionano optical mapping, and also Hi-C data. To achieve long-read data, we do need ultra-high molecular weight or high molecular weight DNA. So at least a 45 kb fragment length, if not megabase length DNA for something like optical mapping. Now, for 70,000 species, that's quite a wide variety of sample types. So this is taxa, tissue types, as well as sampling differences. This provides a challenge. So these are non-model organisms more often than not. So how do you preserve samples when collecting from a field to a lab? The best known method for doing this is sample storage that is flash freezing or ultra cold storage, such as -80 degrees C. However, access to liquid nitrogen or dry ice can be quite a challenge, if not impossible, for many field conditions. On top of this, for some field conditions, even something like refrigeration or wet ice can be challenging. So what do we do then? And so the sample prep group of the VGP set out to answer this question by testing an array of different preservation methods for ultra-high molecular weight DNA isolation. Although we weren't able to test all the possible combinations that might mimic field conditions, we were able to test across 5 vertebrate lineages, so a mammal, a bird, 2 turtles, as well as amphibian and bony fish samples, as well as multiple different tissue types, including muscle, blood, ovary, spleen, as well as isolated red blood cells and whole body. We tested multiple different temperature treatments from 4 degrees C to room temperature and even 37 degrees C. And these temperature treatments, for instance, could be up to a week at room temperature. And then we tested multiple different preservation methods, such as flash freezing, which is considered the gold standard for preservation, as well as ethanol, DMSO buffer, as well as commercial products such as RNA Later, DNA Guard, and AllProtect. After all of these samples were preserved in their different method, we used the agarose plug method to extract DNA. Once we had the DNA, we looked at, of course, the yields that we had from each of these samples, but really we wanted to look at the fragment length also because we're aiming for high molecular weight and ultra high molecular weight DNA. To look at this, we assessed pulse field gels as well as femtopulse traces, and we also looked into Hi-C performance for these samples as well. So to quickly give a summary of what we found that impacted fragment size for these samples, specifically proportion of fragments over 145 kb, we found, not surprisingly, that higher temperature treatments had a lower proportion of high molecular weight DNA in our samples. So From this, flash freezing is still the best choice for your sample. However, interestingly, we found that samples stored at 4 degrees C for up to 6 hours showed no significant difference in proportion of fragments over 145 kb compared to LN2 preserved or flash frozen samples. This allows some flexibility up to 6 hours. Additionally, when it came to different preservation methods as far as solutions go, we found that samples preserved in ethanol showed significantly larger fragment length proportions compared to those stored in DNA guard. Furthermore, when just assessing the pulse field gels, we found that flash freezing and ethanol showed greater fragment length or better results across compared to other samples. Additionally, we looked into the Hi-C performance. So when doing this, we used predictive QC. So looking at the percentage of or proportion of long-range cis interactions greater than 15 kb. We were able to test blood and muscle samples from zebrafinch. And we found that the samples with no preservative that were flash frozen did great, as well as those that had no preservative stored at 4 degrees C for 6 hours. On top of this, we found that samples stored with ethanol at 4 degrees C for up to 1 week also performed very well for Hi-C. So this allows a lot of flexibility when sampling if flash freezing is not an option. So now I'd like to present general guidelines that we found from this study, as well as from all the VGP samples that we've worked with so far. So I can present recommendations based on what type of tissue type per taxa. So something like nucleated blood, such as amphibians, birds, reptiles. We see that blood is a great choice. So this is good for both yield and fragment length more often than not. For something like a non-nucleated taxa, such as mammals, we see that soft tissues would be a great first choice. So think spleen, kidney, liver, and if that's not possible, all of these skeletal muscle is a great second choice. When it comes to the preservation method, though, we see flash freezing as really the best choice still. However, short-term refrigeration for 4 degrees C or on ice for up to 6 hours really has little decrease in fragment length or quality of your sample compared to flash freezing, which is really great because you can afford yourself, a few hours if you can't flash freeze right away after collecting. If that's not possible, midterm refrigeration is also a great choice. So samples stored in 95% ethanol or DMSO solution can be stored on ice or refrigerated at 4 degrees C for up to 1 week with minimal decrease in sample quality as far as fragment length goes. Additionally, there is room if refrigeration is not possible. We've seen success and positive results from samples stored in, specifically blood stored in 95% ethanol at room temperature for up to a week. So we still see traces of high molecular weight DNA. So on top of this, we have a bunch of other different things. So please see our preprint on bioRxiv. And I would like to thank everyone involved in this study and all of our collaborators. And I'm very happy and looking forward to answering further questions on all the things that I haven't been able to go into so far. So thank you.
Hi, my name is Nancy Holroyd and I lead the Tree of Life sample management team at the Sanger Institute. Today I'll be talking about supporting sample flow to feed the growing tree of life. In 2019, the Wellcome Sanger Institute established the Tree of Life Genomics Programme, building on the foundations of the early vertebrate and 25 genomes projects, and under the lofty header of the Earth Bigenomes Project, a handful of collaborative flagship endeavours were launched, notably the Darwin Tree of Life and Aquatic Symbiosis Genomics. Combined, these projects aim to produce chromosomal assemblies of over 70,000 species from across the British Isles and the world. Whilst the Tree of Life is by its very nature richly diverse and complex, and it's for that very reason that we wish to explore its genomic basis, generating chromosomal genomes from that rich diversity has to be tackled in as uniform a manner as possible. High throughput pipelines are required in order to deliver the genomes within our lifetimes. In this talk, I'll give you a flavour of how we're channeling biodiversity into and through a uniform pipeline in order to reveal the genomic wonders of biodiversity at the other end. For the Darwin Tree of Life project, the majority of samples are channeled through the main genome acquisition labs with whom the sample manifest and metadata validation process has been developed. The pipeline supporting the Darwin Tree of Life sample flow from the GALS is well established and robust. Much of what follows in this talk focuses on sample flow from institutes other than these genome acquisition labs. The source of many samples coming into the programme is diverse, not only in location and sample type, but also in terms of the providers. Some researchers will only ever provide one species to the programme, whilst others may submit samples on multiple occasions. It's important that the pipeline is comprehensive enough to fulfil the scope of the grown projects, but that it remains accessible for occasional or one-off submitters. Before talking about the pipeline, it's important to note that the basic recipe for generating high-quality reference genomes assemblies include long read and scaffolding data from, where possible, just one genotype, with gene finding evidenced by transcriptomics data. Critical to the success of the pipeline is the input of high molecular weight DNA. To achieve this, the gold standard has to be samples cryogenically stored in liquid or at -80 as soon as possible after sacrifice to prevent nuclease degradation. Additionally, to support the processing of samples at scale, the pipeline requires all samples to be provided in standard barcoded FluidX cryotubes that can be scanned and minimize human processing error. This is particularly important when handling thousands of samples. It was important to build our systems to be able to support a wide range of partners in providing these gold standard samples where possible. The process flow shown here is the pipeline for sample submission. It starts with the collector onboarding form on the right, which captures general information to enable the sample set to happen. This onboarding form captures key project, partner, and sample information for that sample set, informing the legal and compliance processes and supporting the provision of barcoded tubes where needed by partners. It notifies us of an imminent sample set, and at that point we assign a unique sample set identifier that tracks the progress of the samples throughout. This avoids ambiguity. Many individuals within Sanger, and indeed at the providers' institutes, will be involved in this submission process, and it's crucial that communication and process is clearly linked to a defined set of samples. The onboarding form also provides visibility of samples coming into the pipeline, informing forecasting, particularly important as we scale up. Arguably, the bare minimum information needed to accompany a reference genome assembly is the species name. However, further metadata, such as the original and current sample location, date and method of collection, are required to research governance and wildlife and environmental law officers to verify the samples have been responsibly and ethically sourced. These fields are integral to determining compliance with the Nagoya Protocol, as well as UK and international laws. Yet more metadata are really important for informing the lab and assembly processes, such as the sex, life stage, and tissue type. Sample metadata are recorded in the sample manifest, and data are validated through COPO to ensure the fields are completed according to the standards specified. Comprehensive SOPs support metadata collection, but can be time-consuming for the one-time submitter to digest. So we've generated resources to assist with this and frequently provide guidance and support to researchers to guide them through the submission process. Once samples are approved to be shipped, it's essential that we operate a reliable, safe cold chain to transport samples from source. We've established an excellent relationship with BioCare, who deliver dry ice, dry shippers to our partners and bring samples to Sanger. BioCare manage our fleet of Worthington dry shippers fitted with bespoke shelving to maximise the number of samples. And they'll charge and deliver the dry shippers to locations around the UK within 24 hours. The service has also been used to support sample prep in the field, and we're trialling a portable dry shipper that will fit into a backpack to be used for sample collection and preparation in less accessible locations. BioCare also manage a fleet of dry ice boxes which are reused repeatedly, minimising packaging waste. We rely heavily on a custom developed sample tracking software system, STS, which tracks the full pipeline. On arrival, it's critical that samples are verified and appropriately stored before being passed on to the lab. All tubes are scanned and the contents of each shipment are verified against the manifest to ensure no unexpected samples are included and none are missing. All our freezers and the locations within them can be swiftly registered in our tracking system, enabling fast and unambiguous sample locations to be provided to anyone that wishes to find them. Individual tubes can be moved from one rack to another, allowing us to consolidate partially filled racks and maximize efficiency in storage. Many teams and individuals feed into all this work, and the processes continue to be improved. Our pipelines need to be robust enough to support genomics at scale, but also to be able to accommodate diverse projects, including resequencing for population genomics and whatever the future might bring in terms of biodiversity genomics at Sanger. We're constantly working on improvements and further resources to assist with sample submission. I'd like to thank all of my colleagues and all of our partners that we work with, and it makes this entire endeavour extremely rewarding. Thank you for listening.
Hello, I'm Liam Crowley. I'm a postdoc at the University of Oxford, working on the Darwin Tree of Life project. This talk will be about sample acquisition for genome sequencing. My work is on the Wytham Genome Project, which is part of the Darwin Tree of Life project, based at Wytham Woods, just to the west of Oxford. This is slightly different to other collections on the Tree of Life project because this is all focused within one specific site, which we've called a genomic observatory. Our focus at Wytham is primarily on arthropods and other invertebrates, although we are collecting all different species. We started collections in the summer of 2019. So after just 2 years of collecting, we have collected just under 3,000 samples, which come from just under 2,000 individual specimens. These represent 1,287 different species from 256 different families. So we've already got a pretty good coverage and diversity of samples, including some particular highlights of some more scarce and rarely seen species. Some of these represent new species for the site or even the county. Some of these are due to recent range expansions of these species, but others are just generally not seen very often. And in particular, we found some very rare species, including this silver stilettofly, which we found earlier this year, which was the first time it's been found outside its existing known couple of sites. So the sample acquisition process looks a little bit like this. I'm just going to go through this one by one. Before we can do any kind of collecting or processing at all, we need to have an idea of what we're trying to get. So the first step is species lists. So we have just constructed a list, which is just over 70,000 species, primarily from the UK species inventory and some more up-to-date taxon checklists. Once we have a good list of the UK fauna, we can then identify targets. And this breaks down into two areas. The first is family representative, so 1 or 2 species from every family. And the other is focusing on some particular groups in more detail. So this could be taxonomic groups, or this could be groups of associated species. And there's different criteria for assessing whether a species is a target, but generally speaking, we've gone for the most important, iconic, or interesting species. On other practical considerations about can we get hold of samples, and will there be high quality samples if we do get hold of them. Once we know what we need to get, then we can go out and actually collect the samples using a range of different techniques depending on the samples we're going for. Once we have the live samples in our hand, we need to have a good idea of an accurate species level identification before we can process them. There are different ways that we can identify specimens. The first bunch of specimens we did were mostly on field characteristics, so going for kind of low-hanging fruit, species which we can confidently identify in the field, such as macromoths. There are other methods such as temporary knockdowns and classic microscopy. as well as dissecting out bits of key morphology, such as genitalia. And for some cases, if we can be very confident that they're conspecifics with other species, with other individuals, then we can process one and identify the other in traditional ways. For example, if we know that they are siblings. And then finally, for some species, they may not be reliable external characters that we can use, where we can look into using barcoding to determine species identity. Although this is a very small number of species, all specimens which go through the project are barcoded, but generally speaking, this is as a confirmation of identity, not as blind identification. Once we know what the sample is, we can then get to the stage of processing it. All our processing so far has been done from live specimens using either dry ice or liquid nitrogen to snap freeze and preserve high quality, high molecular weight DNA for long read sequencing. Large specimens are split up into manageable tissue chunks. So, as I mentioned before, a slightly different approach at Whiteham, whereby we are, rather than going out to different areas and collecting to a list of targets, we are intensively collecting and surveying a finite area of one site. There are certain advantages to this method, but also there are some limitations. Generally speaking, the advantages, because we're sampling everything, we have quite a broad taxonomic representation. And we also have the obvious advantage of logistics. For example, having nearby lab facilities to use, we can collect all through the year. We need one set of blanket permissions, and other more niche things such as rearing facilities, for example, to rear out leaf miners and their parasitoids. We also have a good extensive site history for Whiteham. It's a very well-known site with very long-term running experiments. And another advantage which has come to the fore more recently is the fact that we can then, based on feedback, go out and collect further material, either for topping up of specimens which have already entered the pipeline, or the genomes could identify potentially interesting questions, so highlight further species which we can go out and sample from the woods. The limitations, of course, is that because it's one specific site, there's a limited selection of the nationwide fauna, depending on the habitats present at the site and also the region where the site exists. Whiteham's quite good in that it has a wide range of habitats, and it's southeast England, so it's generally got quite a good representation of species. But of course, there are many species which have been identified as family representatives, for example, which would not be present at the site. And just to finish, a couple of case studies highlighting this in practice. So the first of those is hoverflies, for which we have a really good representation at Whiteham, a large number of species. We've already collected 75 species overall from a wide range across the hoverfly family phenology, including some rare species. Hoverflies were chosen because they're species of evolutionary and ecological interest, and because we've got good representation of diverse different life histories, this could potentially throw up some interesting genomic findings. The second case study is on Lepidoptera, specifically non-butterfly Lepidoptera, so moths. We have collected to date 458 species from 50 different families, which is, again, a good selection across the phylogeny, although there are some which we will not find at the Whiteham site. And this really highlights the advantage of the feedback and targeted collecting, whereby we found, for example, in the China mark moths, the first species we collected, we found interesting duplications of Hox genes. Therefore, we went out and intensively targeted China marks and collected several other species to go in and look for similar duplications. That just leaves me to acknowledge all the people who've helped out with sample collection and identification at Whiteham Woods. And of course, this is just the first stage in a long process that before we end up at a published genome. And I'd like to acknowledge all the people downstream, all the hard work that they've put in as well. Thank you.
Hi, I want to thank the organizers who invited me to talk about our recent work, quality control of long-range data such as Hi-C, 10X, and hypertyping. In the Darwin Tree of Life project, we aim to produce reference quality genome assemblies for 70,000 species, including vertebrates, insects, plants, fungi, and many more. In the assembly production, long-range information is very important, and this can be used for scaffolding, base polishing, phasing, and annotation. Here we show three platforms, Hi-C, 10X, and HyperTyping. Hi-C is called chromosome conformation capture. For a pair of Hi-C reads, one end can be mapped to a chromosome location, and the other end mapped to another location. So there is a mapping separation distance. For the longest mapping separation distance can be equal as a full chromosome length. So in a good Hi-C library, majority of the paired reads should be mapped to the same chromosome, and the mapping separation distance should be long and has a good distribution. 10X and the HyperTyping data, they are very similar, and we call them linked reads. With barcode tagging, reads from the same molecule are grouped together, and sometimes we say those reads which share the same barcodes are from a single molecule. You can see here, there are more fragments or barcodes for HyperTyping than the 10X, but number for each barcode for HyperTyping is less than 10X. So that's major difference. We propose 4 metrics for QC Hi-C, as well as 4 metrics for 10X and HyperTyping. First, we're using linked length mapping separation distance, mapping ratio of interchromosome, which means reads mapped to different chromosomes, evenness of base coverage, PCR duplication. So here I list 2 software tools for the users who are interested to have a try. For 10X and HyperTyping metrics, so the same linking length, this we call it molecule length or barcode length. Read density of barcodes, we say M50 reads per barcode or per molecule. Evenness of base coverage, PCR duplication. The same, I also list 2 tools here, SCOF 10X for 10X data analysis, and SCOF HedgeTag for HyperTyping data analysis. Here I show a few figures on OCA library datasets. So you can see here, we show the mapping separation distance, interchromosome mapping, base coverage, and PCR duplication. Here, I also, for the length distribution, I also plotted human Hi-C and the human hummingbird datasets, which previously we have evaluated, which is very good quality. As you can see here, for the 4 OCA libraries, they all follow the trend as human and hummingbird dataset, which is very good news, as you can see here. And for the interchromosome mapping, which is lower than 30%, it's kind of a threshold we use to say this is good or not a library in our case. You also see here the base coverage distribution. So I added human Hi-C, human 10X, and human Illumina short insert size data for comparison. Looks like Illumina still the best, but you can see the clear peak over here. Looks like Illumina is best, and the ORC distribution base distribution also looks good here. For PCR duplication here, kind of 13 or 40%. I think it's good in our case. Another one, I show you a failed case, a library failed case, which is a ladybird. For the Nance distribution, as you can see here, plotted against the human hummingbird, and the trend is not follow the pattern of the human and the hummingbird. The slope is deeper, and as you can see here, it's fast approaching zero, which means a Nance distribution is short. And when you look at the base coverage, we didn't see clear peak here for the two ladybird Hi-C library. You can see here for the red and the light blue. And when you look at the Hi-C map, it's also very messy. It's not very good. So this case, we kind of regard it as a failed library preparation. Here I show you a table. So we have sequenced a number of samples from oak, insect, and mouse, and some fish. So you can see here the PCR duplication here, this column, M50 reads molecule and molecule length and depth. Generally, you can see here also compared with 10x data. So 10x data has higher M50 reads per molecule. And also the PCR duplication is lower than we've seen here for the hypertyping libraries. On the right side, you can see a coverage figure, which I show the oak hypertyping against oak 10x, and also with the human Illumina, human 10x, and human Hi-C. In this figure, you can see here for the for both oak 10x and oak hypertyping, they're all very good in this case. For Nance distribution, so 10x against hypertyping. So you can see here, I show here for this small table here. So average and 50 looks like in this case, oak hypertyping length is slightly longer than 10x, which is very good news. So a brief summary our work looks like the metrics we proposed are working for our QC. Failed or succeeded libraries are in line with our QC predictions. For the 10x and hypertyping metrics, we also see it works. And for 10x and the M50 reads per molecule is notably higher than hypertyping. For hypertyping, there is a relationship between M50 reads per molecule and the PCR duplication. So we are doing lots of tests to sort this out. Also, we are doing more tests to confirm the replacement of 10x with hypertyping. There are so many people involved in this project. Ed and Shane helped me to process data, Joe Wood for annotation, Needs, and Michael, who are making the libraries, Marek, Frank from Germany, who developed the hypertyping method. Thank you also for the audience.
Thank you very much to our presenters for 5 fantastic talks. We received quite a lot of questions and we don't have that much time to get them all answered. So let's get straight at it. So Anthony, Artur Buzinski asked you whether you've tried less concentrated ethanol because the concentrated one might dehydrate the sample and that might be detrimental for the DNA. There seems to be quite some interest in preserving samples with ethanol. So maybe you and Jennifer could comment on sample preservation in ethanol in general.
I don't think we've tried anything less than 95%. But the comment about the dehydration and the DNA quality, I think Jennifer can actually directly answer that based on the experience that they've had.
Yeah, certainly. So we haven't tested extensively lower concentrations of ethanol, but we actually have processed quite a number of samples that are actually stored in 100% ethanol as well as 95%. And it does work exceptionally well most of the time. I think there are some reports of brittleness that can happen for a sample, but for preserving ultra high molecular weight DNA, it works very well.
Okay. What happens, and there are some questions on this, what happens with very small samples, for instance, single insects, very small body size? What would you recommend for those?
Is this for me?
It's for both of you, actually.
Oh, okay. Jennifer, do you want to go first this time?
Yeah, sure. So we've worked with a couple of different arthropods, so single mosquitoes, single arachnids, and they work very well preserved, flash frozen, of course, but also in 95% ethanol. Beyond that, we have not tested.
Mm-hmm. Do you have any thoughts on how your preservation results might be reflected in non-vertebrate samples? This is a question from Andy Griffith to Jennifer.
Yeah, of course. So this is tricky because of the wide diversity of course, invertebrates. So things like the single mosquito and similar samples do work very well in ethanol, and I could imagine also in a DMSO preservation. Other samples, though, I don't want to comment because we haven't tested.
And maybe Nancy wants to say something to that, because Nancy, you commented in your talk on the fact that for Darwin, we expect everything flash frozen.
Yes, absolutely. I think as much as possible that really remains the gold standard, but being realistic and pragmatic in the field when samples can be preserved in ethanol or another solution, and also not having to have that cryogenic cold storage chain, that really opens up some huge possibilities. So it's really encouraging.
Here's a question from Alan Archibald, which was asked to Jennifer, but maybe also Nancy could comment on this. He asked, did you consider the suitability of the different preservation methods and treatments for compliance with trans-border shipments? For instance, compliance with biosecurity regulations?
Yeah, so absolutely. So of course, the flash frozen sample is great, and sometimes that can be tricky. So ethanol is one of our highest recommended preservations. However, that it can also be difficult to cross borders or with different carriers. So that's why we've looked at DMSO for one of the other reasons, and that should be a little bit easier.
Yes, certainly it can have an impact on import or export paperwork that needs to be put together where it has to be documented. And similarly, to have all of the samples flash frozen without having to worry about buffers certainly keeps things simple. But again, being pragmatic, as long as those are documented in the metadata and all of the sample information, then it's possible to get them through. It does add another complication, however.
Mm-hmm. So the last question regarding extractions was asked by Kevin Kokot to Liam, actually, and he asked what tissues you're using for DNA extraction and whether it has resulted in any contamination issues from, for instance, gut contents. Liam, do you want to answer this? I can say something to that as well, maybe.
Sure, yeah. So for all of our arthropod work, we're using whole specimens, mainly because they are so small. Most of the fauna we've collected so far are somewhere between 5 to 10 millimeters total body size. So just to have enough tissue, we're obviously processing the whole. And in terms of gut contacts, that's something that's very important. So far we've primarily focused almost exclusively on adults. For many species, they will do none or very little feeding as an imago, whereas as larvae, which is basically a soft sack of food, so avoiding those where possible. Although we have began testing some larvae, some caterpillars, so we'll see what the extractions look like with that.
If I can comment on this, so we encounter a multitude of contaminants, but also of symbionts and cobionts in the samples we sequence, and it's just one step of the process to identify those and set them aside. So it doesn't make the assembly any easier, but it also doesn't make it impossible. So it's not really our prime concern to get sort of totally clean samples, which is obviously not possible anyway with use sample from the field. Mark Palmada-Flores, Liam, whether you are in touch with taxonomists and how that works.
Yes, lots of different taxonomists. Generally speaking, we work with specialists in a particular group who will come and do intensive collections, depending on their group of expertise.
And last question for Liam. Nancy asked whether there are any or many species that you expected to sample at Wytham, but you couldn't find them yet. And if so, why is that? What do you think?
Yes, definitely. So sorry I didn't have time to say, but one of the first exercises we did at Wytham, because we had a specific site with a long history, is we looked through the list for the national list and identified which species of our target species we're actually likely to find at Wytham. And then we can then kind of guide our collections towards those. And there are some species outstanding on there that we just haven't found for any other reason, that they are just haven't turned up yet.
Thank you. We are out of time, but let me ask one last question to Zemin, because we sort of neglected you, given that you talked last. I apologize. Mark Palmada-Flores asked whether you've encountered any feature in a species that can lead to high C or linked read data showing uneven coverage along the genome, despite good libraries. Zemin, you want to answer that?
Yes, indeed. We see a number of cases in which we thought the library was good. It has long mapping separation distance. However, the evenness is bad. We see particularly fungi and some insects, we see this kind of behavior. Even for ARIMA version 1, version 2, and also like version 2, the mainly improvement is the improved evenness of the reads, which leading to benefit for genome scaffolding, and maybe perhaps in the future, could it be for base polishing.
Okay, with that, thank you very much to all our presenters for your fantastic talks and a really interesting Q&A session. And I'd like to point the audience at the poster session from Specimen to Sequence, where some remaining questions might be answered. There are also mini talks from the presenters, and you can, of course, contact all the presenters over there. With that, thank you very much. Goodbye, and see you at the social at 7 o'clock.